welcome everybody to Grand Rounds, and it's a pleasure, certainly a pleasure to introduce Kaylee Schwertfeger. She's an assistant professor in our department. She was recruited to our department several years ago. I forget how many exactly. How many? 7. 7 years ago, when I was 2. And we recruited her, a combination of the Cancer Center department, to help lead an effort on the tumor microenvironment. That's become an area of intense interest in cancer biology and research from the last several years. What's known now is that it seems to be absolutely required for a tumor to grow and expand and progress, hence the title, which Kaylee's going to talk about her work. She has been extremely successful getting funding in what we would call a thin funding environment. And she has contributed a lot both to the Cancer Center, to the MyCab graduate program. She's assumed a leadership role there, has advised students successfully who have matriculated over the last couple of years, and she's just done an outstanding job teaching. Her research is fantastic, in my humble opinion. And it says here that she's also certified with the Minnesota Board of Pharmacy. So afterwards, anybody wants drugs? I've already got my order in. She came to us from the Baylor College of Medicine, where she did her postdoctoral appointment, and then came here to accept an assistant professor position. So really looking forward to, is that sufficient? All right, very good. Looking forward to having her talk to us about her research. Kaylee.
Okay, thank you very much, Jim, for the introduction. And just to comment on the drugs, that's solely so that I can give drugs to mice after surgery. So if you're in need of about 0.1 mils of buprenorphine, I'm your person. Otherwise, I think the street value of that would be about $10. So thank you for the introduction. And really, I have sort of 2 purposes for this talk. The last time I spoke in front of this group was 7 years ago when I was starting up my lab. So it was kind of fun to go back and look through that PowerPoint presentation. And I'm very glad that things have changed since then. Not giving the same talk as 7 years ago, so that's good. And then the other thing is, when I started my lab, I was trained in sort of mouse models of cancer and really wanted to move my lab into more of a translational research direction. And so I've taken some steps to go in that direction, but what I'm really interested in is sort of fully moving into that direction, which requires really good clinical expertise. So I'm hoping to recruit some interest and some collaborators who might be willing to sort of help me out with the more clinically relevant questions, and I'll sort of highlight some of those really preliminary studies as I'm going through. So to start, let's see, I have no financial relationships to disclose. Everyone kind of gets up here and says, sadly, I don't have these, but I was thinking this probably creates a lot of extra paperwork, right? So I'm kind of glad I don't have financial relationships. And so brief overview of the talk. I'm going to start out introducing you to the tumor microenvironment and the specific aspect of the microenvironment we're interested in, which is inflammation. And then I'm going to give you 2 sort of stories of a couple of the projects going on in the lab, just to kind of give you a flavor of the types of things that we ask. And then I'll talk briefly at the end about a new project that was just funded that I'm very excited about starting with Mike Farrar. So just to introduce you to the tumor microenvironment, this is the primary research focus of our lab. Obviously, tumors don't grow in isolation. They grow surrounded by a complex stroma that's comprised of various types. Oops. Oh, this is, see, I always sort of giggle at the people who do that, and now here I am.
We'll giggle.
Yeah, please giggle at me. So it's comprised of various cell types, including endothelial cells and pericytes, of course important for angiogenesis, fibroblasts, cells associated with lymphangiogenesis, inflammatory cells such as neutrophils, macrophages, mast cells, and others that are somewhat dependent on the tumor type and tumor stage. And in addition, the tumor is surrounded by a complex extracellular matrix. This is sort of a framework of protein and non-protein components that help provide, in a normal tissue, help provide structural support. And we'll talk more about this later in the talk. So it's well accepted in the cancer research field by now that not only does the tumor stroma help support the tumor as it grows, but components of it actively contribute to tumor growth and progression. And so that's what I'll try to convince you of today. So there are a lot of various models around that have shown that stroma contributes to tumor growth. And this one was mentioned to me by Jim a few days ago, which I thought was kind of interesting. This is in Bob Weinberg's most recent cancer biology textbook. And apparently back in the 1960s, the model organism of choice was the human. And there were several groups that were trying to do this. They actually would isolate basal cell carcinoma cancer cells, and they would transplant them onto the same patient, just in a healthy tissue area, and they wouldn't get tumor growth. And then this one group came along, and what they did is they took cancer cells, and then they took cancer cells along with stroma. And they did this transplantation, and they were able to get tumor growth. So I don't know if this is the first specific example of how stroma contributes to tumor growth, but it's certainly the least reproducible. So to introduce you to the inflammatory microenvironment, which is really what we're interested in. You've probably heard the term tumors are like wounds that never heal, and that's because if you look at their microenvironment, they look very similar to what you would see in a wound healing environment. So when you have an epidermal wound, for example, you get infiltration of inflammatory cells driven by production of cytokines and chemokines, and so these are things like macrophages as well as neutrophils. Once these come in, one of the primary focuses of the macrophage is to get rid of pathogens. They phagocytose and get rid of pathogens, but they also do a whole host of other things in this microenvironment, including produce angiogenic factors to promote angiogenesis, as well as proteases and other things that will help remodel the stroma. And so these are all very important for normal wound healing processes. And of course, in a normal wound healing situation, you have mechanisms that sort of tone all this down, and you end up with a completely resolved wound. However, in the tumor environment, you have sort of the persistent presence of this tumor, and this drives sort of what would look like a chronic state of inflammation associated with this tumor, and this is what has been linked to tumor growth and progression. So in the inflammation field, we throw around terms like inflammation and cancer a lot, and a lot of us have sort of different meanings of what this really means. And so just to introduce you to a couple of theories in the cancer field, and that is that you can see inflammation that is induced by extrinsic factors. So you've heard of things like chronic viral or bacterial infections leading to increased risk of developing cancer, such as for the hepatitis viruses. And these are because this chronic inflammation can set up this sort of tumor-promoting microenvironment, and then if you have an oncogenic event occur in one of the epithelial cells, you're more likely to develop a tumor in that type of situation. And then the other concept is that of intrinsically induced inflammation, and this is where oncogenic activation within a tumor cell actually induces inflammation. And so this isn't coming from an extrinsic force, this is coming from the tumor cell itself. And so in the case of breast cancer, which is what I study, this is thought to be the primary driver of tumor-associated inflammation, is that the tumor cells are inducing it themselves, and I'll show you some examples of that today. So as I mentioned, macrophages are very important mediators of inflammation. They're also important promoters of cancer. And so this idea came about a couple of decades ago, and more recently we're seeing more and more studies coming out in breast and other solid tumors, basically showing that the more macrophages you have in a tumor environment, the worse the survival, demonstrating that macrophages are in fact there to help promote the tumor to grow. And so macrophages, again, the tumor-associated macrophage field really took off about a decade ago, and since then we've learned a lot about macrophages and what they're doing both in the normal wound healing environment and in the tumor microenvironment. So for example, macrophages are big producers of inflammatory mediators such as COX-2, which of course is the target of sort of our non-steroidal anti-inflammatory drugs. COX-2 has been shown in many different cases to be tumor promoting. Macrophages also produce chemokines and angiogenic factors, which promote further leukocyte recruitment as well as angiogenesis. They produce high levels of reactive oxygen species, which, of course, in the normal situation is important for removing bacteria and other pathogens. It's been speculated that in the cancer environment, it may actually help contribute to the accumulation of mutations. So not only is it promoting sort of this environment, but they're actually acting directly on the tumor cell to induce further genetic mutations. They're also They're also producers of growth factors and proteases. Growth factors, of course, induce proliferation of various cell types, including tumor cells and fibroblasts. MMPs are important for stromal remodeling and invasion, as well as release of latent growth factors in the stroma. And sort of something I'll come back to maybe a little bit later is one of their primary functions actually in the tumor microenvironment seems to be in promoting immune suppression, and sort of this links these cells to adaptive immunity, and that's sort of the one of the directions that the cancer inflammation field is moving towards. And so, excuse me, so that sort of takes care of the tumor microenvironment and the inflammatory environment. And so the first project I'm going to talk to you about is sort of goes along the lines of this intrinsically induced inflammation. And what I'm going to show you is that activation of oncogenic growth factor receptor signaling in tumor cells induces its own sort of pro-tumorigenic inflammatory response. And so we're interested primarily in growth factor receptor signaling in cancer because in many cancers, aberrant activation of growth factor receptors and their downstream signaling pathways has been shown to promote tumor formation and progression. Probably the most well-studied of these would be the ERB family, so EGF receptor, which is involved in a wide variety of cancers, as well as ERB2, which is, of course, involved in breast cancer. Another family of interest that we focus on is the fibroblast growth factor receptor family. This is a fairly large complex family comprised of 4 FGF receptors, which can also be alternatively spliced into different isoforms. And there are 22 FGF ligands which can bind to these receptors, induce dimerization and activation of downstream signaling pathways, and I'll show you those in a minute. In terms of breast cancer, there have been a wide number of studies showing that FGF receptor activation is important in breast cancer. It's been known actually for quite a while that there's amplification, particularly of FGFR1, which is located at chromosome 8p12 and is amplified in about 10 to 15% of human breast cancers. Interestingly, you also see amplification of ligands such as 3, 4, and 19. This is located on 11q13, which is also the cyclin D1 amplicon, and interestingly, these two are often amplified together, so that provides a potential mechanism for increased FGF receptor activation. Another mechanism through which FGFs promote breast cancer is that cancer cells will start increasing high levels of FGF ligands, which can then feed back in an autocrine manner to activate signaling. Several years ago, there were a number of activating mutations identified in FGF receptors, and so we anxiously awaited all the sequencing data and were a little disappointed to find that in breast cancer, we really don't see many FGF receptor mutations. They are there, but just at very low levels. So it seems like FGF receptor activation in breast cancer is primarily driven by other mechanisms. Importantly, FGF receptor inhibitors are currently being tested in phase 1 and 2 clinical trials for breast and other cancers. And so, when I started these studies back as a postdoc, all the grant reviewers say, "Ugh, FGFs aren't important in cancer." And so now we've sort of moved to the point where they're now being tested in clinical trials. So we will see how those pan out. So of course we work on breast cancer, and the big question we always get, of course, is which subtype of breast cancer is FGF receptor signaling involved in? And so I'm not going to go into the details of the molecular breast cancer subtypes for these talks. I'm just going to focus on the breast cancer subtypes that are driven by presence of receptors. And so, of course, we have the hormone receptor positive cancers, ER, PR positive, as well as ER and PR negative. And most of the FGF receptor amplifications are actually found in ER positive and specifically luminal B breast cancers. And this confers resistance to endocrine therapy. So there's a lot of interest in using FGF receptor inhibitors in patients that develop resistance to these therapies. In the HER2 positive cancers, the evidence isn't very strong, but there is some experimental evidence that FGF receptor activity also confers resistance to HER2 positive treatments such as trastuzumab and other targeted therapies. And then in triple negative breast cancers, this seems to be where ligand overproduction is important. So they found that in up to 60% of triple negative breast cancers that lack ER, PR, and HER2, that these express overproduction of FGF ligands. And so it seems, so understanding FGF receptor mechanisms has importance for not just a single subgroup of patients, but could span a wide variety of breast cancer patients. And so when FGF binds to its receptor, this induces dimerization and activation of all these pathways, which I'm not going to go through. The point of showing this slide is that these pathways have all been pretty well worked out. We have a very good knowledge of what's happening directly downstream of FGF receptor signaling. So these are pathways involved in things like proliferation, survival, migration, et cetera. What always cracks me up about, I'm a signaling person, so I love these diagrams, but what always cracks me up about them is that it always stops here at the nucleus with target genes. Okay. And so this is really what we're interested in. We're a little less interested in these specific signaling molecules and more in what are the target genes being regulated by FGF receptors. And specifically, we propose, and we've seen in our studies, that a lot of these target genes are soluble factors. And so these would be things that are secreted by the cell to go out and affect things like inflammation, changes in extracellular matrix, angiogenesis, and fibroblast activation. And so that's really what our focus is understanding what these target genes are and then how they impact the tumor microenvironment. And so to do our studies, we use sort of a variety of human and mouse different cell lines as well as mouse models. And I want to introduce you to one that's a little bit unique to our lab. It was developed in my postdoc mentor's lab, and and so we're really the only ones who use this model extensively. And this is an inducible model of FGF receptor activation. So as I mentioned, FGFs bind to the receptors, induce dimerization, and then activation of all the pathways I showed you. We wanted to make an inducible model for reasons I'll tell you in a minute. And to do this, we actually removed the ligand binding domain completely so that it doesn't bind to endogenous FGF ligands. We targeted it to the plasma membrane with a myrstillation sequence, and then we added a dimerization domain. And what this does is it binds to the synthetic dimerizer that we get from Clonetech, and when this is lipid soluble, and so this gets into the cell, induces dimerization and activation of downstream signaling pathways. And this model was generated over 10 years ago, and we've shown, I think, very conclusively that this does activate all of the same downstream signaling pathways that endogenous FGF receptor does. So the advantages of inducible models, particularly in the mouse modeling field, is that we can precisely control oncogene activation in mice. And what this allows us to do is better mimic some somatic oncogene activation observed in cancer cells. So we were sort of, forever, we were using sort of these transgenic models that were overexpressing something from birth. And whether that really represents a somatic mutation that occurs in an adult tumor cell is somewhat questionable. So this allows us to model this a little bit better. The other advantage, and I'll sort of point this out as we go along, is that we can study cell type specific oncogene activation in co-culture models. So in the tumor microenvironment world, of course, we have to think about other cell types. And so we do a lot of co-culture type models. And if you just throw FGFs in a co-culture model, you're not, and you see effects, you're not really going to know which cell type it's affecting. And so this allows us to specifically activate FGF receptors in tumor cells and then look at the effects that has on sort of surrounding cells. And so we use this model. We have this in cell lines, and you'll see some of that data as we go along. We also generated a mouse model, so we can drive this inducible FGF receptor in mammary epithelial cells using the mouse mammary tumor virus promoter. And this is just a mouse that hasn't been treated with the dimerizer that induces activation. This is a mouse, a duct from a mouse mammary gland. And you see it's a nice layer of epithelial cells. This is the adipose stroma. When we activate FGFR1, we see a very reproducible increase in these sort of budding lesions. And we tried calling it lateral budding at one point, and the reviewers didn't like that because these aren't normal buds. They're not actually surrounded by myoepithelial cells like a normal alveolar bud would be. So we call this just aberrant budding. of some sort. One comment, the last time I did show this slide in my talk 7 years ago, and one of the comments I got from one of the pathologists was that this actually doesn't look that different from human budding, and so why is this abnormal? And in the mouse, this is actually not something that you ever see unless you start to see these things when the mice are pregnant, but in mouse models you don't really see this. So this is aberrant for a mouse. If we keep treating the mice, we'll start to see formation of alveolar hyperplasias. You can see a blood vessel right here. This is a very nice model, actually, for angiogenesis. And then if we treat the mice for long enough, we'll eventually get tumors. But our focus really is on these early stages of tumor initiation. And so how did we get into the inflammation field? So the first experiment I did actually as a postdoc is I took these mammary glands and I ground them up and I did microarray analysis. That was sort of the gene analysis approach of choice back in those days. Now it would be RNA-seq. But we got a whole host of cytokines, chemokines, and things involved in inflammation. And since then, we've gone back and shown in this is a cell culture model that we use of mammary epithelial cells expressing this inducible FGF receptor. And we've shown that epithelial cells actually produce a lot of these things. And I'll get back to that later. But these data made us start thinking about inflammation and how FGF receptor is inducing inflammation. And so we started looking at inflammatory things, and this included macrophages. And what we found is when we induced FGF receptor activation, we saw a very dramatic recruitment of macrophages to the stroma surrounding these sort of hyperplastic lesions. So we do see some macrophages. This is F480 staining here. In wild-type animals, they're just sort of normal tissue macrophages, but you can see a dramatic recruitment here. These remain associated with these lesions even out 4 weeks, and then we see, if we look at tumors, we see macrophages associated there. And so we did some sort of genetic techniques. I won't go into details, but we depleted macrophages from these mice. And this is whole mount analysis. These are mammary glands taken from mice and then stained with hematoxylin. And when we activate FGFR1, you can see in comparison to these normal ducts here, these are terminal M buds and they're distending ducts, subtending ducts. You can see these nice budding formations occurring. When we deplete macrophages, and again, I'm not going to go through all of this, just take my word for it that they're not there, we see a reduction in budding. Both here you can see that compared to this terminal M bud neck, and we also see that when we look at the ducts as well. And so this suggested to us that macrophages were actually promoting tumor initiation in this model. And so this is sort of the generic model we think about in the lab. We activate FGF receptor signaling in an epithelial cell or a tumor cell. This produces secreted factors or soluble factors that can then both recruit macrophages and then activate them. The macrophages then produce more soluble factors that can either act back on the epithelial cells, and this is the project that I'll tell you about, as well as do other things that I mentioned previously. And so some of the co-culture models that we use are described here. And this is sort of a complicated model. We even get confused in the lab when we start talking about doubly conditioned media. So I'm going to walk you through it. So we take our epithelial cells that express our inducible FGFR1. We throw the dimerizer on them, and then we collect the soluble factors that are produced by the cells. We take those, we put them onto macrophages. This is a macrophage cell line. We also use bone marrow-derived macrophages to confirm our studies. So we take these factors, we put them onto macrophages, and then we take this, what we call doubly conditioned media, and we ask whether it can promote epithelial or tumor cell migration. And so the data are shown down here. If we just take these soluble factors, we can in fact induce epithelial migration. And this makes sense. The FGF receptor activation is producing lots of things that are promoting macrophage migration. But then if we take this doubly conditioned media, we can enhance this migration, suggesting that macrophages, in response to the factors produced by these tumor cells, can enhance migration and thus theoretically tumorigenesis. The other kind of cool model that we've set up is a primary 3D co-culture model. And so for these, we actually take primary mammary epithelial cells out of our mice. We put them into recombinant basement membrane matrix, matrigel, which is comprised primarily of laminin. And these form these nice little three-dimensional structures. So this is just normal epithelial cells not exposed to dimerizer. When we activate FGF receptor, these things get bigger. They don't look invasive, they don't look scary, they just get bigger. But then we can throw, I don't know, can you see this? And if you throw macrophages in here, the macrophages actually sort of coalesce around these structures. And if we look closely, we can see that the macrophages are associated with sort of epithelial cells that seem to be invading into the matrix. And so this was our model for studying macrophage-induced epithelial cell invasion. And we can quantify this here. We see a significant increase in the percentage of invasive structures when macrophages are present. So these are kind of the co-culture models that we use to define the mechanisms that we ultimately then want to go back and test in our mouse models. And so in order to study these effects on migration and invasion, one of the things we looked at was the production of chemokines from macrophages. Macrophages produce high level of chemokines in general. Chemokines, of course, produce induced directed cell migration. Oops, let's see. Okay. So we took our conditioned media from the FGFR activated cells, put it onto macrophages, and then we looked at chemokine-related genes. So this was just sort of a targeted panel of genes. And by we, I mean Laura, who's a postdoc in the lab. She did all this work. We identified a host of various chemokine-related genes, and we don't need to, these aren't important what they are, but several of these were induced in the macrophages. When we focused specifically on chemokines, which chemokines were induced, we found that 4 of these, CXCL1, 2, 5, and 7, were induced. And these are all of interest because they bind to the chemokine receptor CXCR2. This is a G-protein coupled receptor, which once activated by ligand induced a whole host of more signaling pathways, so lots of signaling pathways going on here. Including ERK, P38, et cetera, and then this can induce primarily chemotaxis in the cells. Chemokines, of course, are very important for leukocyte recruitment, but their effects on tumor cells are not quite as well understood. And so we were particularly interested in this interaction, and we hypothesized that production of these ligands could act through CXCR2 to promote migration and invasion. And so we did some validation studies just showing again, that macrophages produce high levels of these chemokines, including CXCL1, CXCL5. Importantly, we also wanted to make sure our epithelial cells express CXCR2, otherwise it wouldn't have much effect, and they do express decent levels of CXCR2. And then we can take a CXCR2 inhibitor, and we can ask whether this can inhibit macrophage-induced epithelial cell migration. And we find that, that it does in a dose-dependent manner. So this suggests that these chemokines are in fact directing epithelial cell migration. And we also expanded our studies. As I said, we validate a lot of our studies in models of endogenous FGF receptor signaling to make sure they translate. And again, we looked at MCF7 cells, which respond nicely to FGFs as well. They also induce CXCL1 and CXCL8, also known as IL-8, which gets a lot of attention in the cancer field. We looked at a panel of cell lines at CXCR2 expression. MCF7 cells seem to express high levels of CXCR2. So we did this assay, and again, we see the same thing, a nice reduction in response to the CXCR2 inhibitor. Then if we went back and looked at our invasion assay, again, this is what I've already showed you, the macrophages sort of coalesce and induce invasion. When we throw the CXCR2 inhibitor in here, the macrophages are still there, and they're still sort of recruited around these structures, but we really see very few invasive structures. So we're not getting rid of the macrophages, but we're inhibiting their ability to induce invasion. And so this is the model sort of based on those studies. We have tumor cells, which are producing soluble things, and I haven't talked about that, but I'll come back to that at the end, which recruit macrophages. These secrete these CXCL 1, 2, 5, 7, 8 chemokines, which act through CXCR2 to promote migration and invasion. So this is sort of reminiscent, if we start thinking about more human context, this is somewhat reminiscent of this idea that ductal carcinoma in situ can transition into invasive breast cancer, which I realize is sort of a controversial topic in the breast cancer field. However, we think that what's happening is if you see macrophages are recruited to these lesions, they might help the tumor cells to invade and possibly become an invasive breast cancer. And then the other question is, are these interactions targetable in breast cancer patients? So what I didn't mention is that CXCR1, CXCR2 inhibitors are available. They're actually being tested in clinical trials, initially for transplantation rejection, but now they're actually being tested in breast cancer clinical trials as well. And so there are potential mechanisms for targeting these interactions in breast cancer patients. And so I was talking to my friend. I have a collaborator down at the University of Kansas, Fariba Beybad, and she's an expert she's developed all sorts of models to study DCIS invasion. And she said, well, you really should look at macrophages in DCIS that are associated with microinvasion. And she had samples that she could send me, so she sent me some human DCIS samples. I don't remember the definition of microinvasion. I believe this is also a somewhat controversial topic in the breast cancer field. So this is where it'd be really nice to get some clinical expertise in sort of understanding this. But basically, and Polly in the lab did this stain, and so she stained for CD68. And so basically what we found was that in pure DCIS with no what was called microinvasion, we really didn't see many macrophages associated with the stroma. There is some staining in here, and we've talked about whether that's real. There's also a lot of controversies going on here. There's a lot of controversy too whether macrophages are actually in DCIS lesions. That was sort of a hot topic in a recent meeting we went to. So that there may be some macrophages within the DCIS, maybe not, we don't know. But we did find, as we were looking through these DCIS with micrometastasis sections, we saw a lot more inflammation associated with these lesions. And of course, along with inflammation, we see more macrophages in the stroma. So these are the tumor cells here. But we see a lot more macrophages in the stroma of these lesions. And so we're really interested in pursuing this idea that macrophages are actually promoting invasion of DCIS or early stage lesions, if not DCIS. Okay, so that was the first story. The second story, I'm going to switch gears a little bit and move away from macrophages, but I will come back to them because everything we do has to go back to macrophages. And I'm going to talk about the inflammation-associated remodeling of the extracellular matrix and how this contributes to tumor growth. And so when we think about inflammation, we think a lot about infiltrating immune cells, but we don't think quite as much about changes in the extracellular matrix. And these are actually quite important for the inflammatory process. So this is a fairly complex slide, but basically the idea is that you have this sort of framework of extracellular matrix. This is comprised of proteins like laminin, collagen, fibronectin, et cetera, as well as non-protein components like hyaluronan, which I'll talk about more in a minute. And what happens is tumor cells or other cells in the wound healing environment secrete proteases. And these proteases start to chew up this extracellular matrix. And this converts sort of larger molecular weight components into these small fragments, which are actually bioactive. They're called matrokines. And they can elicit different effects on different cell types compared to the high molecular weight forms. And so these bioactive fragments, and particularly hyaluronan, which I'll talk about in a minute, can then go and actually act on infiltrating leukocytes, such as macrophages. This stimulates the macrophages to produce more MMPs, and this feeds back into the extracellular matrix. It also induces, as I just showed you, cytokines and chemokines that can act back on the tumor cells. And so you set up this sort of positive feed-forward loop and this could partially explain the reason that we see chronic inflammation in the tumor microenvironment, or chronic inflammatory responses in the tumor microenvironment. So when I first started here, I was working across the hall from Jim, and every time I showed him these models I had, he would say, "So have you looked at HA? Have you looked at HA? Have you looked at HA?" And for a couple of years I said no. And then finally I looked at HA.
Just made me go away.
Yes, but it had the opposite effect. And so HA is actually a very interesting component of the extracellular matrix. It doesn't get a lot of attention, but we're going to change that, right? That's the plan. It's actually synthesized as a very high molecular weight polyanionic carbohydrate. And my feeling is that it hasn't got a lot of attention because it's not a protein. It doesn't come up in people's gene arrays and things like that. And everyone's so focused on those. So that's what I think. I think it's very important. The high molecular weight HA maintains physical properties and hydration of tissues. It's very important for structural support of tissues. HA fragmentation or degradation by things like hyaluronidases and reactive oxygen species that are induced during wound healing are a warning sign of tissue damage, again promoting this inflammatory response. If you look at HA levels in cancers, we see that elevated levels are associated with cancers and actually are prognostic. And HA, as I'll show you, is important for tumor growth, survival, and therapeutic resistance. And so just 1 second on the hyaluronan life cycle. It's actually synthesized by the aptly named hyaluronan synthases, which sit on the plasma membrane of a cell and extrude hyaluronan to the extracellular matrix. Once it's in the extracellular matrix, it can then bind to receptors. CD44 and RAM are the two I'll talk about today. And these sit on various cell types, bind to HA and induce even more downstream signaling pathways. It can also bind to ECM proteoglycans, and these can sort of maintain their functions in the extracellular matrix. We had Tom White out here a few weeks ago who talked about Versacan and had some very nice data showing that. And then finally, HA can be internalized, and this mechanism I don't know much about, but it can also be degraded, as I mentioned, by things like hyaluronidases as well as reactive oxygen species. And this is, as I mentioned, important in inflammation in general. So in breast cancer patients, people have looked at levels of HA. And basically what they found is that if you have very strong levels of HA in the so this is in the stroma, not necessarily in the tumor cells themselves, this is associated with reduced survival. And if you look in the parenchymal HA, again, not as strong of an association, but you still see a reduction in survival. So the more HA, the worse it is, basically. And so we looked at HA in our models, and this is staining for HA using a biotinylated HA binding protein. And so in the wild type mammary gland, we do see some HA surrounding the epithelial ducts, which makes sense. This is a normal component of the extracellular matrix. When we activate FGFR1, we see a nice recruitment or induction of HA in the stroma surrounding these aberrant budding lesions. And we've also looked in various FGF receptor-dependent tumors, such as our cell line that stably expresses our FGF receptor. We see plenty of HA associated with these tumors. HS578 T cells are a triple-negative breast cancer cell line that we can grow in mice as well, and we see plenty of HA in these tumors as well. So the question is, of course, HA is there. What is it doing? Or actually, first, we asked what cell types express HA. HA can be expressed by fibroblasts, tumor cells, et cetera. So we wanted to see if tumor cells could induce HA expression in response to FGFs. So we looked at a panel of breast cancer cell lines. Again, these are our mouse cells, and then these are 3 human breast cancer cell lines. And in all of them, in these first 3, we did see an induction of HA. So this is HA concentration in the media. This is an ELISA-based assay. And we see nice induction of HA in the media in MCF7 cells, which are an ER-positive cell line. HS578s have fairly high levels of HA basally, but we can bump that up a bit with FGFs. And then if we take BT549 cells, which have very high levels of autocrine FGF receptor activation, and we treat them with an inhibitor called PD-17307-4, which is an FGF receptor specific inhibitor, we can tone down the production of HA by these cells, linking FGF receptor activation to HA production. And so we actually, a couple of years ago, we went through Bionet and we had them make us a very large breast cancer tissue microarray. We have over 200 patients. We have normal DCIS, invasive cancer, and then inflammation, focuses of inflammation on this tissue microarray. And so we started doing some of these studies. We kind of found out that doing all the IHC and all the scoring and all this stuff ourselves is fairly challenging. And so this is another area where I'm sort of looking for, help, clinical expertise, et cetera. But we are able to stain human tumors with phospho-FRS2. This stands for fibroblast growth factor receptor substrate 2. There are no good antibodies for staining FGF receptors in breast cancers or in human samples. So this is sort of a marker of activated FGF receptor signaling. We see moderate to high levels of FRS2 activity in about 80% of human breast cancers, so that's pretty high. And we can also stain with HA, and so I'm not trying to denote any sort of correlation here. I'm just showing that we can do the staining. We see plenty of HA in the stroma, and then in some cases, in this one, for example, we do see some HA staining within the parenchyma as well. And so we're interested in trying to maybe correlate FGF receptor activity to HA in these sections, although we're not quite there yet. So next we wanted to ask, if HA is there, what is it doing functionally in the tumor? And so we started out by trying to define how HA was being produced. So we looked at the different hyaluronan synthases, and we found that HAS2 expression was induced upon FGF receptor activation. If we knock down HAS2 with an siRNA, we're able to effectively reduce both basal and FGFR-induced levels of HA production. And I'm not showing it, but we've also validated these in lots of other cell lines, and we know this is true across a variety of cell lines. We were then able to block HA synthesis using 4-MU, which Tom White talked about a few weeks ago. This is an inhibitor of hyaluronan synthase. And so we did a couple of in vitro studies looking at scratch wound closure. This is just a measure of migration, where you measure the ability of epithelial cells to cover a wound. And FGF receptor activation induces migration, and this can be inhibited to some extent by inhibiting HA production. We also looked at proliferation. Again, we see something similar. FGF receptor activation induces proliferation, and this is inhibited in the presence of 4-MU. So then we went in vivo. So of course, these things usually work in vitro. The question is whether this actually corresponds to in vivo studies. And what we did is we provided 4-MU to mice via oral gavage. And this is our MMTV FGFR1 model. So the control mammary glands look fairly normal. When we activate FGFR1, we see lots of budding in the epithelium. When we throw 4-MU in there, we still see some budding, but it seems to be reduced. And so we can quantify this by counting the percent of severe budding, which I won't go into. And we see a significant reduction in severe budding in these 4-MU treated animals. And we also see a corresponding reduction in BRDU. So this was interesting to us and suggests that this increased production of HA does in fact contribute to initiation, at least in this model. So then we wanted to ask, well, what happens if we try to target HA in an established tumor, which would sort of more mimic the clinical situation. And in this case, we took HS578 T cells. We threw them into 3D culture. So again, throwing them into matrigel, this recombinant basement membrane matrix. There's lots of growth factors, there's serum, there's all kinds of things around. And these cells form these kind of blobs in 3D culture, not the nice, pretty structures I showed you earlier. But what we did is we took these cells and we threw 4-MU on them, did absolutely nothing to proliferation. PD did nothing to proliferation. And that actually surprised us, because at least in 2D culture, when we don't have serum around and it's a very contrived model, these cells are actually FGF-dependent. So clearly in sort of this 3D environment where you have lots of serum around, they lose that dependence on FGFs. But when we throw the two of these in there together, we see a significant reduction with an exclamation point, apparently, in proliferation. And so this was actually interesting, because this suggests that on their own, these inhibitors are doing absolutely nothing. But when we combine them, for whatever reason, and we don't know yet, they actually work together. They work synergistically. And so we've tried to do these studies in vivo, and they were not as exciting as we hoped. But then Tom White came out, and he was talking about 4-MU in mouse models, and he was showing their conditions. And so they actually provide the 4-MU in the food at really, really high concentrations, much higher than we were doing. And so I think we're going to revisit these studies in vivo using his protocols. because it sort of worked, but I think if we could enhance our protocols a little bit, we'll be able to show this in vivo as well. And so to go back to our model, basically what we've shown is that FGF receptor activation induces expression of HAS2. And what I'm not showing you is a whole lot of work that has gone in to understand the mechanisms through which HAS2 is induced. And so this is actually not a direct activation of HAS2. It actually requires induction of other soluble things, which then act back on the epithelial cells. I'm not showing that because of time, but this is a little more complicated than I'm denoting here. But the end result is that HAS2 is produced. It goes up to the membrane and extrudes HA into the extracellular matrix, which we can measure by ELISA. And this promotes things like migration, proliferation, and I didn't show you, but it also promotes therapeutic responsiveness. You actually see reduced response to chemotherapy in these cells as well. And so in the first scenario, in the in vivo model that I showed you, that first scenario, that's sort of a very FGF receptor dependent model. And so in that case, we're able to, so for example, if we had an early stage tumor that was sort of dependent on this single pathway, you'd think we could block FGF receptor activation. This would reduce HA production and reduce tumor initiation. In a later stage model, where you've had FGF receptor activation on for a long time, you have lots of accumulation of HA in the microenvironment already, just blocking FGF receptor activation may not do anything at that point because you've already significantly changed the microenvironment. And so this is the case where we probably need to block both. And so that would be my explanation for the results in the 578 T cells. And so this is something that we're very interested in exploring, this sort of combinatorial-based approach for targeting the tumor cell and changes in the microenvironment. So I want to give a couple slides just on sort of, this has all been published, I want to give you a couple slides on current directions and sort of the ways that we're thinking about this now. So when Tom was out here, he showed all these data with 4MU. And then he made a point that, well, this probably isn't, 4MU really isn't going to be a drug in people because we do need HA synthesis to occur normally. So we probably won't be able to use this in people. And so really the next step is to understand the mechanisms through which HA is acting and figure out ways to target those rather than targeting HA production specifically. And so HA has several receptors onto which it can bind, and two of these are CD44, which has gained a lot of attention in the cancer stem cell field, as well as RAM. RAM stands for receptor for hyaluronan-mediated motility, I believe. So obviously it binds to HA, and this helps promote migration. And so these are both actually have been linked to tumors, and so we've been sort of focusing on these two receptors. They're expressed on tumor cells as well as a wide variety of other cell types as well. The HA-CD44 interactions are important for, as been published, inflammation, T cell recruitment and activation, tumor growth and metastasis. And then HA-RAM is important for tumor cell migration, oncogenesis, tissue injury, and repair. And so you would think that targeting these things might have a significant impact on tumor growth. And so we've started doing some of these studies. You can get blocking antibodies that actually inhibit the HA-CD44 interactions. And we've thrown some of these in our models, and we see a reduction in migration, suggesting that HA is at least partially acting through CD44. Jim is actually working with Barry Finzel in chemistry to develop small molecule antagonists of the CD44 HA interactions, so we're hoping that our models will be good use for testing those types of approaches. We've also been working with Eva Turley up at the University of Western Ontario, who's developed a whole host of things to target the HA-RAM interactions, and so we're hoping to start using those in our model as well to sort of tease out which receptor interactions might be important or not in our model. I also have HA-TLR up here as well. So these are toll-like receptors, and as promised, I want to bring us back to macrophages. So in the tumor microenvironment, of course, HA, as I said, HA can act on tumor cells themselves directly through CD44 and RAM. But in the microenvironment, in the inflammatory microenvironment, as well as in the tissue microenvironment, HA gets degraded into these small HA fragments. And these fragments can bind to and activate macrophages primarily through activation of toll-like receptors 2 and 4. Macrophages also express CD44 and RAM, so this could get a little complicated. But the idea is that HA is activating macrophages. And the interesting thing in our model is that, again, this is the aberrant budding that we see in our model. This is the enhanced stroma, and I'm not showing it here, but the macrophages are all accumulating within the stroma. And incidentally, this is the stroma that has all of the HA. So when Tom was here, he had beautiful pictures showing co-localization, red and green, of macrophages and HA, and so I really want to do that. But I think that this suggests that the macrophages are being recruited to this HA-containing stroma. And so those are some more mechanisms that we're interested in studying. So those were the three sort of main things I wanted to touch on. Again, we have various projects going on in the lab. These were just a couple of them. And I want to end by giving you an idea of some of the ways that we're trying to go in the future. And this is based on a grant that was just selected for funding. This was an R21 that was a response to the NCI Provocative Questions. So I don't know if you guys have seen these, but the NCI came up with a whole list of questions that they think are really important, things that have been difficult to study or not focused on or whatever in the field. And they've set aside money for this. So this is a nice mechanism because we know there's going to be money in this mechanism, unlike just the regular submissions. And so this particular question was what molecular and cellular events in the tumor microenvironment, for example, the local immune response, determine if a tumor at the earliest stages of malignant transformation is eliminated, stimulated, or made indolent. And so we were excited about this because we thought that our model would recapitulate the earliest stages of malignant transformation. So we were very excited about this opportunity. And yeah, I should have switched that slide. I asked Mike Farrar if he'd be a co-I on this grant because I really need some immunology expertise. I'm good with epithelial macrophage interactions, but moving into adaptive immunity is a whole new field for me. So hopefully Mike will help me make that transition. And so the reason for this is that what's coming out more and more in breast and other solid cancers is that if you look at the composition of immune cells in the microenvironment, it's actually, this can be prognostic. So if you have, and this actually makes sense if you think about it, if you have high levels of CD8 T cells, which are of course cytotoxic, low levels of macrophages and low levels of CD4 cells, patients are going to do better. So this is a more favorable tumor or inflammatory immune response environment than patients that have lots of macrophages and low levels of CD8 T cells. And so the inflammation in cancer field is really starting to move in this direction. What's sort of driving the generation of this type of tumor pro versus anti-tumor environment? And that's sort of the basis of my interpretation of the basis of that provocative question. And so as I said, we thought our model would be a nice one to study this. So obviously we're getting an environment that's permissive for tumor growth. But we thought we could use this model to try to identify these the mechanisms driving this. And so, as I said, when you get an initiating event, whether it's activation of FGF receptor, whether it's p53 mutation, whatever sort of initiating event you have, your cells start secreting stuff, and you get lots of soluble factors. We've analyzed our cells to death. We know by proteomics, gene array, we know what they're making. And if you look at what they're making, it's not an overt pro-tumor type of phenotype. You see lots of things that could easily be promoting an anti-tumor response as well. And so we think there's a balance here. So you need the proper balance of anti versus pro-tumor responses. And if it's shifted in the anti-tumor direction, your immune response is going to get rid of it. If it's shifted in the pro-tumor direction, they're going to grow. So basically, the first part of this grant is to take a fairly targeted approach because we do know we do know a lot about these factors, and we're going to take a targeted approach to try to identify which are the critical sort of balancing factors that lead to the generation of a pro versus anti-tumor immune response. So then once you have these factors, then of course you have macrophages. Macrophages will respond to whatever these factors are, and then they'll produce their own pro or anti-tumor factors. So this is already getting sort of complicated. And so again, if we can sort of modify these soluble factors, we may be able to figure out which ones are important for bringing in these pro-tumor macrophages versus macrophages that might be actually anti-tumor. And then to get even more complicated, of course, then we start seeing whatever component of T cells that come into the microenvironment or are sort of in the peripheral tissues as well. And these, of course, secrete all of their own stuff. And so you can quickly see how complex this gets. And so really it's the balance of all of these things that determines a pro versus anti-tumor immune signature. So in this proposal, as a in addition to the targeted approach, we're also going to use some sort of larger scale genomic approaches to not necessarily look at the tumor cell, which is what everybody does in the field. We're actually going to look at different subsets of these immune cells and sort of try to tease out which pathways are involved in this whole process. And the ultimate goal of this project will be to develop novel strategies, of course, for enhancing anti-tumor immunity at early and other stages of transformation. And so this was an R21 that was selected for funding. It was supposed to start yesterday in true NIH fashion. We're still waiting for the NOGA, so hopefully that'll come soon. So I will finish, and I want to acknowledge the people in my lab, of course, the people who actually do the work. I just sit in my office and put together pretty pictures. Laura is a postdoc who worked on the CXCR2 story. And is working on some other macrophage-specific projects now. Nick Brady and Polly Chuntova are graduate students in the lab. Polly worked on the HA project along with Laura. And Nick is doing some macrophage-directed studies right now, as well as developing some novel models of FGF receptor-driven tumorigenesis, additional models that we can use in our studies as well. And then various past members contributed to these studies as well, particularly Ron, TJ and Yoon Gil. Of course, Jim McCarthy, because it's his fault we're looking at HA. However, I have to say thank you because he's introduced me to a lot of people in the field. I've been to Canada to meet people. He's really opened up a lot of doors for me, and so I'm very excited about this project. Andy Nelson in the department, who I bother on a constant basis about with my random pathology breast cancer questions. Mike, with whom actually we have another project that Mike's been helping us out with, and then I'm excited to get these studies started as well. I mentioned Eva, who works on RAM and HA, and then Fariba, my friend down at University of Kansas, and then funding. And also my lab is located in the relatively new CCRB building. If you guys haven't been out there to see it, you're welcome to come out, take a tour. I'd be happy to show you around. It's a very beautiful building, and it's really not that far away, I promise. So I will be happy to take any questions. Yes?
Kaylee, I really enjoyed that talk, and I'm going to ask you a question that didn't really relate to your research.
Okay.
It related to your first slide on macrophages.
Yep.
I gather you're very enthusiastic about macrophages, and that's what I want to ask.
Yes.
You alluded to this question in that first slide. What I want to ask you is, to what degree does the microenvironment contribute to tumor cell phenotype modification as it metastasizes?
To tumor cell phenotype modification as it metastasizes, and specifically with regard to macrophages?
Well, or any part of the microenvironment.
So the macrophage field actually was born out of this idea that macrophages, and these are really beautiful studies. These are studies done by John Pollard and John Candelas, or Jeff Pollard, John Candelas. And what they've done is if they've taken sort of tumor models and they've looked at how macrophages affect invasion into blood vessels and subsequent metastasis. So they have this really aggressive tumor model, and if they completely deplete macrophages from it, they get no metastasis whatsoever. And so they've actually shown that, and they're just beautiful images. John shows these at national meetings, where they have a tumor cell that's green and a macrophage that's red, and they kind of line up in a line. And you can see them proceeding towards blood vessels, and then the macrophage will sit there. The tumor cell will enter the blood vessel, right where that macrophage is sitting. And so if you get rid of macrophages, you don't get any entry into blood vessels. And so from the macrophage standpoint, they seem to be critical, at least in certain models, for tumor metastasis. And actually for that reason, so these studies came out, a while ago, and there are now clinical trials actually targeting macrophages in breast and other cancer patients targeting CSF1 receptors. So I know that, you know, from a macrophage standpoint, they're absolutely important for invasion into blood vessels. As far as other parts of the tumor microenvironment, I think there's quite a bit known about invasion, but I know most about macrophages.
But they're involved to some extent also in mutations, genetic mutations in the tumor cells?
So that's an interesting idea that I think hasn't necessarily been proven yet.
Oh,
So
okay.
the idea is that if you produce lots of reactive oxygen species, you're going to be inducing mutations in the tumor cell. So if you have a tumor cell that's sort of not fully blown yet and sort of waiting for a couple more genetic mutations, they might help that out. That hasn't been proven. That idea has been around a while. That hasn't been proven yet.
Okay.
Yeah.
You know, you mentioned that a normal body, and that the reviewer said that they're not surrounded by epithelial
Yeah.
cells. I don't know how mouse histology of the breast tissue works, but in the human, you can no longer consider that an incital lesion. So that would be my microinvasion, in fact.
Yeah. So, and we're actually we're looking at that literally right now. We have observed in the past that you lose the myoepithelial cells pretty early in this model, which is interesting because you're right. You know, DCIS sort of the definition is that you lose the myoepithelial cells.
Well,
And I mean the definition of right. Yeah. Yes, exactly. Yeah. So you have the and then you need to lose it theoretically in order for them to become invasive. So we're actually thinking about, and this is, you know, sort of in the thinking process, is sort of using this as a model to understand how the myoepithelial cells are going away, because we're very interested. We have no idea how they're going away. We just know they are.
also when they're going away.
And when they're exactly. Yeah. So it's probably going to be a little earlier than in humans, but you know, we can still sort of glean out some of the mechanisms, I think, from that.
Very nice.
Thanks. Mike?
So in the first part of your talk, you told us a lot about what the macrophages are producing it as acting on the tumor cells.
Mhm.
But you didn't tell us much about what's bringing the macrophages in.
I did not. So we actually have a couple of, we've looked at a couple of chemoattractants. FGF receptors activation pumps out a ridiculous amount of osteopontin. Osteopontin is a fairly potent macrophage recruitment molecule. So in vitro, we've looked, you know, we've been able to block osteopontin and show that macrophages are, you know, no longer are being recruited. We got the osteopontin knockout mice. This was like the big heartbreak of the lab. That didn't work at all. So it's possible that something's compensating. We also, we did publish a paper on CX3CL1, which is also, it's a monocyte, inflammatory monocyte chemoattractant. And we did show that if we block CX3CR1, the receptor, we're able to inhibit macrophage recruitment as well. And so we think there's probably multiple things. You know, we focused osteopontin, you know, in all of our arrays, osteopontin is through the roof. And so we kind of focused on that one. I think there's probably multiple things that are bringing them in. Yeah, it's been a little tough in vivo, which makes me think there's a lot of redundancy. Yeah, Robin?
Are tumor-activated macrophages themselves capable of inducing dysplasia or inducing carcinogenesis? In other words, do you wind up with a field effect maybe where you've got tumor cells here turn on macrophages that then have some sort of a paracrine signaling effect and induce badness in the epithelium down the road?
Yeah, and that's another good question. Maybe we can talk about these sort of conceptually, because we have a lot of interesting stuff in the lab in terms of things that are happening to normal mammary epithelial cells, and how maybe a lot of these things are kind of, you know, even though the normal epithelial cells don't have the FGF receptor or whatever, oncogenic activation, that a lot of these effects are inducing that. And so I've actually thought about writing grants and things along those concepts. I don't have a good answer for that. I would guess probably, but we haven't.
DCIS did some studies about 10 to 15 years ago looking at if you take neoplastic stroma from breast cancer cells and you put the epithelial in there, you can hit the non-neoplastic epithelial
Yeah.
to mimic the ablation.
And that's kind of actually, a lot of the things we're modeling are like that. So we're looking at effects of like macrophage factors on actually normal epithelium. And so we are sort of modeling that in vitro, but in vivo, we don't know.
It's like you're sort of inducing a little
Yeah.
field effect.
Yes, exactly. Which I think is really cool, actually. So, yeah.
In regards to your study
Yeah.
fibroblastic stroma. So some people believe that that type of DCIS is more likely to have microinvasion and a
Okay.
micro-DCIS. I think that's interesting. And then the other thing I was thinking about is all these patients that are getting neoadjuvant chemotherapy.
Yeah.
So the tumor bed has all these elements in it. So why is that good? Presumably in a patient that has a complete pathologic response where they just have a ton of fibroblasts and macrophages and lymphocytes in their tumor bed versus somebody that has a partial pathologic response who still has some residual tumor cells, but the surrounding stroma looks very similar. I wonder if that microenvironment is truly different or not.
You mean in the patients with the chemotherapy?
Right.
Yeah. I don't know.
So neoadjuvant is
Yeah.
really good chemo prior to surgery.
Yeah, it's a really
Some people have a complete
see
response.
the microenvironment's different between those that
Yeah.
have a complete pathologic response versus partial.
Yeah, yeah.
Sounds like they're good studies for you to work on.
Yep. Very cool. I would love lots of opportunities. Yeah, that's exactly why I presented some of the preliminary stuff, was I would love to talk about these types of projects in a more sort of clinically relevant manner. Absolutely. Jim?
Anybody else
No.
have questions?
Oh, Jim has a question.
And so we've talked about this, Kaylee, but
Yes.
you also, I think, have data to suggest that the microenvironment can actually aggravate the phenotype, say, of a basal type carcinoma. You just want to comment on that?
Yeah, so we do have some data. So I didn't get into the molecular subtypes of breast cancer. Breast cancers have been profiled again to death, and particularly triple negative breast cancers have been at least molecularly broken down into several, it's like 7 or 8 subtypes now because they're very heterogeneous. And there's one particular subtype we're interested in, the Claudin-Low subtype, which has high levels of inflammatory molecules and is typically associated with macrophage infiltration and things like that. And we've at least found, Nick's been working on this in the lab, and I was going to present it but didn't because of time. But basically what we found is that FGF receptor activation actually drives the inflammatory and as well as other parts of the Claudin-Low phenotype. And so we're particularly interested in that phenotype because of its link to inflammation, and we think that FGF receptor is actually driving that part of the phenotype.
Are there other questions? If not, thank you so much.